Hello, everyone.
Hello,
My name is Mina Rowe Johnson. I'm an assistant professor in biochemistry,
and my name is Julio. I'm a fourth year in Minas Lab.
and we're going to go over some background slides for the paper that you'll be reading for class.
Yeah, so we chose this paper because we felt it was a pretty good representation of a cellular process that incorporates a lot of the things that we've been talking about, both in this unit and in prior units, and shows it in an in vivo model. So giving kind of a different view from a lot of the stuff that's been done previously in our unit, which is more focused on in vitro. um, and really to start thinking about how is it that you start to answer some of these questions in vivo, where there are a lot more nuances and complicated environments and factors that you need to take into account that you don't necessarily need to take into account during an in vitro study.
Yeah, and we recognize that maybe many of you may not be familiar with this model, both the C. elegans model, but also tissue invasion. And so we want to go over some of the background on cell migration and tissue invasion to set the groundwork for this paper.
Yeah, so cells breaching the basement membrane is used pretty ubiquitously across the human body, really to carry out a variety of cellular processes across, again, like a variety of tissues, right? So it's something that's pretty ubiquitous. It's something that a lot of cells do, not just cancer cells, but it also is used by cancer cells. It's what allows them to kind of break out from the primary tumor and really start metastasizing in other processes that cancer uses to invade the human body.
Yeah, so if you think that the clump of cells that make up the tumor, that oftentimes just having a primary tumor doesn't really result in death of a patient, but actually its ability to break through matrix barriers. So that's sort of denoted maybe by this red line and some of these gray squiggles. The cancer cell can get out, invade through this tissue, and then enter circulation. And even the entry into circulation requires a breaking of a basement membrane to get past the outside walls of the vasculature into the bloodstream. So this ability of cells to breach basement membranes is a really critical process in metastasis.
And so what's kind of cool about breaching the basement membrane is that it's a great example also of cellular plasticity and the role that it plays in this process. And so cells are very plastic, and that means that they can adopt or adapt accordingly to a lot of different factors or environments around them. And a great example of this is adapting through multiple forms of migration, each with like different characteristics to navigate these microenvironments. So like here we have some examples of a few different types like amoeboid cell migration, mesenchymal cell migration, collective cell migration, and cells will transition between these different states accordingly depending on what they need to navigate their environment. And transitioning through these states also means that there's a transition in terms of the amount of, for example, like cell adhesion that they might use through the extracellular matrix, uh, breaking down the extracellular matrix using either proteolytic or non-proteolytic forms of degradation, depending on the um, type of migration that they're using, and also just the amount of contractility and like shape that they adapt, right? So it's really cool because they're able to use these different forms of migration to really navigate distinct tumor microenvironments. And what's one of those forms of migration that an ECM degradation that this paper kind of focuses on is this idea of like more mesenchymal migration and the degradation of the ECM, right? And so when we're thinking about breaching the basement membrane, and when Kelly et al. Is thinking about it, it kind of started from this idea of how cells are able to degrade the extracellular matrix that Mina just talked about in the previous slide to really start this process of metastasizing and really start to migrate away from whatever primary tumor or wherever they are to start. Yeah, and so in order to degrade the ECM through a proteolytic fashion, so via cleavage, cells often use matrix metalloproteinases. And so when you're thinking about an MMP, um, this right here is the part of the protein that is within the cell. So you have the cytosol, the transmembrane, and then all of this is found outside the cell. So the catalytic domain that will help uh, cleave the extracellular matrix, all of it's extracellular. And so MMPs have been shown to localize to embedopodia during cell invasion. So as the paper kind of talked about, embedopodia is basically like these protrusions that come out that allow the cell to kind of sense its environment, sense the extracellular matrix, and really start to get a sense for like, okay, what's around me? What do I need to cleave? What do I need to get rid of? And overexpression of these MMPs is implicated in cancer cell invasion. So it's been shown that when these are overexpressed, uh, cells are able to invade more easily, and cancer cells take advantage of that to really start to invade uh, through the basement membrane and start to metastasize. And so this is an example kind of showing cells just kind of chilling in like an extracellular matrix or a 3D matrix. And as they go along, they start to migrate, they start to degrade the extracellular matrix. So this is an experiment in this paper where they had, or an example of them using MMPs to physically degrade the basement membrane.
Yeah, so you can imagine then that if a cell needs to navigate through this really dense extracellular matrix environment, that it'll leave little tunnels behind as it chews through the matrix to make a path for itself. But in addition to degrading matrix and kind of clawing away at whatever's in your path, there are forms of migration that are also protease independent. And so these will be cells that maybe they'll face a matrix where they can't chew past it. So instead, they squeeze past it. And so they don't now need to degrade the matrix, but it's almost like if you had a balloon where you squeeze one side of the balloon and then it like protrudes out on another side and another side. Is in more of an amoeboid form of migration to get through these barriers. And this would be an example that Julio mentioned earlier with the different forms of migration that cells will take based on their environment of cellular plasticity, because the cell will get to where it needs to go and it will take on different forms of migration to be able to get to their destination.
Yeah, and it's alluded to in the paper, right? It was shown previously that for cells that were invading, if you're thinking about, oh, okay, cells degrade the ECM in order to invade, then we just need to knock out the components that allow it to invade. So knock out or inhibit the MMPs, right? But it's been shown that even when that's the case, like saying, cells can adapt to a different form of migration to still move the basement membrane, and that's a great example of cellular plasticity. And so there is some controversy if MMPs are actually required for basement membrane invasions. Um, it also doesn't help that a lot of the in vitro studies that have been done early on can't really recapitulate the ultrastructure of the basement membrane, right? There's a lot of nuances and complexities of that microenvironment that you just can't capture in an in vitro system. And so really, in order to get a better sense for how cellular plasticity works for basement membrane breaching, it's better really to transition to an in vivo model where a lot of these nuances and environmental factors are still intact. And so the question is, how do you observe cell invasion in vivo? And while there's multiple ways that you could approach this question, the paper that we focused on, Kelly et al., use C. elegans.
The humble nematode.
Yes.
So C. elegans is an invertebrate model, but is a really ideal model to understand cell and developmental biology. Here's an example of an adult nematode, C. elegans. It's about 1 millimeter long. It's a hermaphrodite, and therefore it can produce its own embryos. And actually, you can see the little embryos inside of its body that then gets laid on the dish. It has great genetics. The life cycle is very short. And so genetics is very easy and rapid. And as this image nicely illustrates, it's optically transparent. The fact that we can even see the embryos inside of its parent already tells you that it's transparent. And when you look at these embryos individually, you can look at the individual cells within an embryo. So because of that, a lot of work looking into the cell biology of development in C. elegans uses fluorescent proteins for live cell imaging, and certainly the paper, the Kelly et al. Paper takes advantage of this approach too. So just to orient you, since some people might not know the sort of the C. elegans nomenclature, this is a lift from figure 1 of the paper, and what these series of alphanumerics mean is, I guess we can start with this one first. This is a ZMP promoter driving GFP expression. So this greater than sign means that it's a promoter. That means any cell that has the ZMP1 promoter active will make this cytoplasmic fill of GFP. This is great if this promoter is specific to a cell type of interest. In this case, it just happens to be specific to anchor cells. And so you can use it to tag and mark anchor cells in the animal. But perhaps you want to look at localization of a protein inside of a specific cell. Then it would make sense to do a fusion protein. That's denoted as these double colon, where you have ZMP2 fused to GFP. So this is now the full length ZMP2 that is fused to a GFP molecule such that you can visualize where ZMP is localized specifically in cells that express ZMP. So in this case, this shows the actual localization of ZMP3 in anchor cells. So throughout the paper, you'll sometimes see the greater than sign. Just remember that's a promoter driving whatever is downstream. And sometimes you'll see the double colon, which is a fusion protein. So that's usually a functional protein that has a GFP tag on it that lets you know where that protein is localized.
Yeah.
Yeah.
Yeah, and another thing that's really great about C. elegans is it's really easy to do, like saying like do a lot of genetic screens on it, and so it's really easy to knock down genes using techniques such as feeding RNAi, which is basically you have bacteria that have double-stranded RNA that's relevant to your or double-stranded DNA, sorry, relevant to the genes that you want to knock out. You plate the C. elegans on those bacteria, and that will help. That will basically knock out those genes through RNA interference. And it's great because you can do that with like a library, or you can do it for single genes if you have like a specific gene in mind. And so it's something that's really, really easy to do, really efficient. You literally just plate them on the bacteria and then you're good to go.
Okay, so the model that is used in this paper then is anchor cell invasion. And we're just going to give a quick introduction on what anchor cell invasion is and why it's important for this organism to put things into context. Okay, so there should be, yeah. If you look at an adult C. elegans nematode, as we mentioned before, they have both male and female gonads because they're a hermaphrodite. And so they can fertilize their own embryos. Those embryos then get deposited through the vulva. If that vulva doesn't form properly, it's infertile. The animal will die, essentially. So it's a really like tightly regulated process because it's highly selected for. Like fidelity of this process is highly selected for. So if you look at a side view of anchor cell migration, this pink line denotes a basement membrane. And that basement membrane separates out the anchor cell, which is labeled in green, with these underlying cells that essentially make up the vulva of the animal. So if you now flip this view 90 degrees, and you're looking at the belly of this cell where it's making contact with the membrane, here's what it looks like, which is essentially a sheet of basement membrane in magenta. And you can just see the hint of the anchor cell kind of on the other side in green. And it's because the anchor cell hasn't yet breached through and poked a hole through that basement membrane. After invasion then, so we're now back at the side view, you can see that there's a break in that basement membrane because the anchor cell has formed a protrusion that has degraded that matrix to now interact with the cells below it. So if you were to flip that view 90 degrees, you see that there's basically a hole where that anchor cell has now pushed through, degraded it, and now is interacting with the cells underneath. Once it interacts with the cells underneath, it then signals to those cells. So what's shown here is a schematic of the anchor cell in green. So that would be here. Here are the underlying cells underneath the basement membrane. So those would be these light blue cells. And you can see that once the anchor cell makes contact with those underlying cells, it sets off a signaling cascade, which is a gradient that specifies and differentiates those cells such that they form the vulval precursor cells and they go on to make the organ. So it's this ability of the anchor cell to degrade the matrix and contact the cells underneath it that allows for proper formation of the organ in the animal.
Yeah, and so it's great to investigate this in C. elegans because it's a good example of an in vivo, or it's a good in vivo example of how cells are able to invade through the basement membrane in a physiological environment, right? And like in the like was saying, in like the I guess like what you would call like the wild type version of this process where it occurs through degradation, right? You can really start to see and investigate how is it that cells are able to degrade the basement membrane.
Yeah, and a lot of the signals in cell biology of this process is also recapitulated in mammalian
Mhm.
systems. So cancer cells do essentially the same thing, which is punch a hole through a matrix to be able to get out of where they are currently into new space. And a lot of the screens that this lab, is the Dave Sherwood's lab at Duke, have done have been shown to also be true in mammalian cancer cells.
Yeah, and so in case you haven't read the paper yet and you're going through the background first, we're not going to ruin exactly what happens in this paper, but Kelly et al. Really is a great example of some of the stuff we've talked about, which is the idea of cellular plasticity for this specific process and what happens, for example, in an animal that can't use matrix metalloproteinases or can't use degradation of the ECM to break through to that next cell layer, right? And so, yeah, we really think this is a great paper in terms of starting to investigate first, how is it that cells are able to kind of adapt to different circumstances that they might be in? In this case, how is a cell able to adapt to not being able to degrade the basement membrane, but still needing to break through? And it's also just a great example of how you can investigate these questions in vivo. Like Mina was saying, at the end of the day, it's not something we can really do in humans. We can't test anything in humans. So using other model organisms that allow you to investigate these processes that are physiologically relevant to things like cancer, it's really a powerful tool and something that really a lot of people, especially nowadays, really look for when you're thinking about what do I want to publish, what do I need to include in papers. An in vivo model is always like a great thing to have and something that if you can pull it off will definitely go a long way.
Yeah, and as you read this paper, but really any paper, be asking yourself sort of what is the rationale?
Mhm.
What is the question? As you lead into a figure, what is the question that they are trying to answer? What tools are they using to answer the question? And do you interpret the results in the same way that they do? And if you don't, what would you want to see instead? It's something that we'll be focusing a lot on during the class, in addition to the findings of the paper. And it's great training for when you enter your thesis labs and as you're preparing for capstone.
Yeah.
Okay, so enjoy the paper.
I will see you
Looking
guys in
for, yeah,
class.
looking forward to a good discussion.
Okay, that was